Hi everyone, this lesson's an introduction into protein synthesis. I want us to start with an example of two pugs. Here on the left you can see Captain Wrinkles has fawn fur, meaning there's parts that are dark and parts that are light, whereas Admiral Wrinkles, no color at all. Why is this happening? Well, somewhere in the DNA of Captain Wrinkles there's a gene, a sequence of DNA that codes for a protein. And thinking of fur color, we're thinking about the pigment melanin. Melanin is what gives fur its color. So what's the story with Admiral Wrinkles? Well, there's got to be a gene that's either turned off or missing for melanin. The reason I bring up this as an example is it demonstrates why it's essential for us to understand the pathway of information in a cell, meaning how does the information that codes for the proteins in the organism go from the DNA to the proteins that make up its structures, because that's going to make up the cells in the organism, their unique characteristics, and that combination of cells is what's going to make up the organism. This flow of information is known as the central dogma of biology, and it's stated pretty simply. DNA goes to RNA, RNA goes to protein. This occurs through two distinct processes. To go from DNA to RNA, that process is known as transcription, and it's going to occur in the nucleus. Makes sense, that's where the DNA is located. To go from RNA to our protein product, we're going to do a process called translation. Hopefully you remember when learning about cells that proteins are made at ribosomes. So let's start with our first process, transcription. In our nucleus, we have DNA. We have one copy. Every cell in your body has the exact same DNA. Yet, cells like heart cells and skin cells are completely distinct. Why? Well, it's going to come down to what genes are read. When we go through transcription, we're going to take that one original DNA blueprint and make copies of it out of a type of RNA called messenger RNA. That messenger RNA is going to be made again and again and again. We can make multiple copies. This is transcription, but why do this? Why make mRNA in the first place? Why not just send DNA out to make a copy of a protein? Well, there's a lot of advantages to doing this. One is it keeps DNA safe. DNA is in a nucleus for a reason. That DNA needs to be protected. If we damage that original blueprint, for the rest of that cell's life, it's going to carry that damage forward. It only has one copy. Making copies of it keeps that DNA safe and untouched. Another reason is we only have one, so it'd be better to run off a bunch of copies. If I need to make a bunch of different copies of a protein like melanin, I can just run off mRNA copy after mRNA copy and use that to increase the amount of protein that I produce. So to go from DNA to RNA, what we're going to need to do is unwind our DNA double helix. That's going to be unwound by DNA helicase, and we're going to need to make an mRNA transcript. To make that mRNA transcript, an enzyme called RNA polymerase is going to come in and build a complementary mRNA strand to the gene, the DNA strand being read. When we open up the DNA double helix, there are two strands, and we want to make mention of both because we have a different sequence on both. The bottom strand, the one that the RNA polymerase is building off of, is called the template strand, whereas the strand that remains open is called the coding strand. These names might seem odd, but they serve a purpose. For example, here we can see a coding strand of ATCG. And a template strand, the strand that RNA polymerase is building off of, is TAGC. When we make our mRNA product, this is what we're going to get, building off of the template strand AUCG. Notice how similar that is to the strand that's not being replicated off of the coding strand. The coding strand is ATCG. The only difference there is U because mRNA doesn't have thymine. So we call the strand we're not building off of the coding strand because that is the information we're coding for with our mRNA strand built off of the template. We run into a problem here. You have hundreds of genes. Each of them code for different things, some for things like eye color, others for height. How does RNA polymerase know where to go to find the beginning of the gene and where it ends? To do that, there are some structures built into your DNA. One is a region that we call the promoter region. The promoter region is the site where RNA polymerase binds right before the beginning of a gene. It indicates the start of a gene. Often these regions are referred to as the ta-ta box because they're always the same sequence of TA, 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 and that redundancy just makes it easy for RNA polymerase to find where to attach to begin transcription in front of the gene. Another region we have upstream or before the gene is something called the enhancer region. The enhancer region is like an on-off switch for the gene. It's upstream from it and it can enhance or speed up transcription as well. So now this diagram is a more accurate representation of how genes are dispersed across your DNA. We have an enhancer, then further downstream we have the ta-ta box where RNA polymerase is going to attach, indicating the beginning of a gene, and then the gene itself, the sequence of DNA that codes for protein. To turn a gene on, molecules called transcription factors are going to attach to the enhancer and the ta-ta box. Once those transcription factors attach, that will then facilitate RNA polymerase to come, attach to the promoter, and build an mRNA copy. So after the gene's turned on and RNA polymerase builds that mRNA complementary strand, we're now left with what we call the premature mRNA transcript. This carries the information of the coding strand. The dilemma we have is this premature transcript is made of two different regions. One is called exons and the other is called introns. Exons are the regions that are going to be expressed. They have the actual code for the gene, what we want to make our protein product. Introns, on the other hand, are regions that don't code for any protein. Oftentimes they're called junk DNA. These are the in-between sections that we don't want. If we were to read this mRNA as written, it would have information in the exons followed by gibberish in the introns. So, problem is, we need to cut those introns out. To do that, we're going to use SNRPs. SNRPs are small nuclear RNA that are able to find introns and cut them out. SNRPs will often work together in groups called spliceosomes. These spliceosomes will go up to the beginning and the end of the intron, cut out the intron, and glue together the exons. This now will give us a mature transcript, a transcript that's made only of exons that are expressed. And there's quite a chunk that's cut out. In this example, you can see we started with premature mRNA that had 10,000 bases. Now we're down to only 1,000 bases with our mature transcript. A note about this process. Something else can occur here post the transcription processing called alternative splicing. The way the SNRPs are going to cut out the introns and glue together the exons can be done in a different order. In this example here, you can see they're spliced so that we have one section that just has the green gene and another section that only has the red gene instead of splicing them together. This is one of the many ways that your genome is able to diversify what ultimately is expressed. Now, before we finish this process, there's one more problem that transcription needs to overcome. That mRNA is going to need to go out into the cytoplasm to be read by a ribosome to produce protein. Cytoplasm is a dangerous place. We don't want our mRNA transcript to get damaged along the way. So to fix that, two things are added to our mRNA. The first is a GTP is added at the 5' end, and this is called a 5' GTP cap. This is going to provide energy for that mRNA. When it goes to the ribosome, it's going to need a little bit of energy to get the process going. On the other end, we add something called the poly-A tail. The poly-A tail is just an extension of adenines. This is there for protection, and it also determines the lifespan of the mRNA. Add only a couple A's, that mRNA is going to disintegrate rather rapidly. But add quite a few A's, and that mRNA can be read again and again to make more and more proteins. And that's the process of transcription. Would you be able to recite every single step that happened? Hopefully. Just to be sure, here's a video to review.
DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes, transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation.
Let's move on now to translation. You saw with transcription, the DNA inside the nucleus is going to be copied by RNA polymerase and will make multiple mRNAs. That mRNA, now that it's mature and been processed, is ready to go to a ribosome where it can be read to build a specific protein. To do this, we had our strand of DNA, we made an mRNA transcript. Now we're going to be able to read that mRNA and translate it to the amino acids that make up a protein. There are 20 total amino acids, so this creates an interesting math problem. How do we code for all 20 amino acids when we're only dealing with 4 letters in the DNA alphabet: adenine, uracil, guanine, and cytosine, dealing with the mRNA transcript, so uracil instead of thymine? Well, if we read one base at a time, so one base code for one amino acid, that only gets us a total of 4 aminos. So let's propose that it reads it by 2 bases, that AU codes for one amino, GC can code for another amino. Well, if you do the math on that, that only gets you 16 codes. We need 20. If we read these bases by 3, though, we end up with 64 possible codes for amino acids. And this is why mRNA is read in triplets called codons. 3 letters are going to code for one amino acid when reading the mRNA transcript. This has resulted in us being able to determine every single codon that codes for every single amino acid, and you can see that here in the amino acid table. All life uses the same codon system and the same codes for the amino acids that make it up. Why is there so much redundancy in this code, though? If you notice, I can have up to 4 codes for one amino acid. Seems a little unnecessary. Well, this is to protect against mutations. DNA can become mutated when picking up factors from its environment or from mistakes in DNA replication. If one letter at the end changes, oh well, probably will still code for the same amino acid and not make a difference because of this code redundancy. I do want you to notice that there's one codon labeled start. That's the codon that initiates translation. The ribosome knows to start building once it hits the codon AUG. There are also 3 codons that operate as a stop codon. These stop translation. The ribosome is going to start with the start codon, read in triplets, and then stop whenever it hits a stop codon. Please know that the start codon does code for an amino acid, methionine, whereas the stop codons do not code for any amino acid. So how are we going to match up these codons? Well, there's another form of RNA called tRNA. tRNA, or transfer RNA, is going to carry amino acids one at a time. To make sure it's carrying the accurate amino acid, the amino acid that matches the codon on the mRNA, tRNA has a triplet system that's known as the anticodon. These are three-letter triplets that are complementary to the mRNA codon. You can see with the first one, AUG's complement is UAC, and the tRNA carrying UAC only carries one amino, methionine. A little more information about tRNA. Here's a 2- and 3-dimensional image of what it looks like. tRNA at the amino acid end is able to attach to one and only one specific amino acid. Structure always determines function. And on the other end, we have the anticodon that will complement and match up to the mRNA codon to make sure it's dropping off the correct amino. tRNAs are reused again and again. So after they drop off an amino, they're going to need to have another amino attached. The enzyme that causes that to happen is known as aminoacetyl tRNA synthase. So we're now ready to look at ribosomes. Ribosomes are where proteins are going to be built. Oftentimes, mRNA will go through a series of multiple ribosomes, making up a structure called a ribozyme. That's just multiple enzymes reading the same mRNA transcript to continuously build multiple proteins. A reminder that ribosomes, they're actually made of a type of RNA called rRNA that's made by the nucleolus, and they have two subunits, a large one on the top and a small one on the bottom. Now, ribosomes have 3 unique regions in them, and those regions are going to be a great showcase of how translation works, how we can read that mRNA transcript to build a specific protein. Starting on the right-hand side, the first site in the ribosome is known as the A site, or the aminoacetyl tRNA site. This is the site that's going to have a tRNA attach, and you can see that in the GIF there, and wait to be added to the middle of the ribosome to build a polypeptide chain. That next site is known as the P site. The P site is where tRNA is going to be holding the growing polypeptide chain. Our goal here is to build a protein. Proteins can be hundreds of amino acids long. And notice how it's moving like a ribbon on a tape with the GIF on the bottom left. It's going to start in the A site and then move over to the P site, and that P site is holding the growing chain. Our last site is the E site, or the exit site. This is where the tRNA, now empty because it dropped off its amino acid in the P site, is going to escape and go and have another amino attached where it can be reused again. These 3 steps are often summarized in a different way. Sometimes the first step is referred to as initiation. This is when the ribosome attaches to the mRNA and that start codon is read. After that begins elongation, and this is a series of tRNAs entering, adding to the growing chain, escaping, and this happens again and again and again so that polypeptide can continue to grow. Our last step is termination. This is when the stop codon is read, stopping the entire process. Do you think you could now describe the entire process of protein synthesis, how we go from DNA to RNA to protein through transcription and translation? Well, I'm going to have us watch a review of translation. Let's see if you remember.
How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3-letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down one codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell.
And that's the story of the central dogma of biology. I hope this was helpful, and I'll see you next time.